Good evening, children. Welcome to another practical assignment for today. We will do practical number 6 under section C of slide preparation. And the practical 6 is germination of pollen grain in a nutrient medium. Till now, from the beginning, we have completed 3 flowers under taxonomy, that was section 1, China rose, sweet pea and petunia. That completed the entire section of your writing assignment on taxonomy. After that, I have started with slide preparation first, and in my previous video, I gave you practical 4, that was TS of a hydrophyte stem, and number 5, that is TS of a xerophytic leaf, that is nerium. Under the same section, we are going to do practical 6, germination of pollen grain in a nutrient medium. Alright, so with today's practical, let us start with the description in short before I read the actual things to be written. The working is very simple and very interesting. The requirements are less. All we need is a special type of a slide, and this slide has a cavity in the center or a depression in the center. We call such a slide as the cavity slide. Now what we do is, in a beaker, we will make a solution of 15% sugar solution, right? 15% sugar solution, and add a pinch of boron, right? And add a pinch only of boron, right? And then mix it uniformly. So this becomes our nutrient solution. Once again, the nutrient solution is 15% sugar solution and a pinch of boron that is added in the same. The same, after proper mixing, will be transferred by means of a dropper into the depression of a cavity slide. Maximum 1 drop would be enough, or if the need be 1 to 2 drops. Now in the same depression, what will happen is that we have to place some pollen grains. Pollen grains of some available flowers like lilium, Catharanthus roseus or antirrhinum. We'll tap the pollen grains into the same and cover this with the cover slip. That's all. We will leave the setup for a few hours and observe. So as I told you, children, the working is very simple, interesting, and remarkable results are seen with this experiment. So when the school reopens, you will be really liking the experiment to conduct because of the simplicity and remarkable results. Alright, so let us see what you have to write now in the file work. Object: to prepare a slide for germination of pollen grains in a nutrient medium. Pollen grains, it would be in a nutrient medium. Apparatus: 15% sugar solution, a pinch of boron, cavity slide, dropper, cover slip, beaker, and pollen grains from available flowers like lilium or Catharanthus roseus or antirrhinum. These are 3 different flowers, children. So any of this which is available we'll be using. Most probably we'll use Catharanthus roseus. So this is requirement. Coming to the next heading is procedure. Prepare 15% of sugar solution and add a pinch of boron. In the brackets you will write, this solution helps in germination of pollen grains. Next point, put a drop of this nutrient solution in the cavity of the slide. Third point, tap the anthers from the freshly picked flowers, break them and dust the pollen grains in the nutrient medium. Fourth point, leave it as such for few hours. And the last point, put a cover slip on the slide and leave for observation. So what would be the observation after a few hours? After a few hours, the cavity slide shows numerous germinating pollen grains in various stages of development. Pollen grains are unicellular and haploid structures. They possess a bilayered pollen wall, the outer exine and the inner intine. Exine is thick and ornamented, whereas the intine is thin and smooth. So I'll just show you this diagram from a theory book which you have done in the lesson Sexual Reproduction in Flowering Plants. The structure of pollen grain goes like this. The outer exine is ornamented, right, and discontinuous, whereas the inner intine is smooth and continuous. So wherever the exine is missing, like here it is absent, here is discontinuous, here is missing, and here. So this region, it is called as germ pore, where the pollen tube will be germinating. So germ pore is actually the germination pore for the pollen tube to emerge. Alright. So the nucleus also, if you revise, the vegetative or tube nucleus and the generative nucleus. You can see the bigger is vegetative or tube nucleus, which goes as a torch bearer at the tip of pollen tube, right? Like this. The tube nucleus is at the tip of pollen tube, and the two male gametes. How have they arisen here? The two male gametes are formed by the division of this generative nucleus. As the name tells, they divide and generate two male nuclei, right? So that was just a theoretical part required for the same. So I'll read it again. The exine of some of pollen grains will be seen to have bursted, and intine has come outside from the germ pore in the form of pollen tube. And the last point, fifth number, the protoplast of pollen tube contains two nuclei. Out of them, the larger is known as tube nucleus, whereas the smaller is called as the generative nucleus. So just now I told you that the larger, you have seen it also, is the tube nucleus, and the smaller is the generative nucleus. So that will be all to write, and the last heading would be result. The above experiment demonstrates preparation of a slide of germinating pollen grains in a nutrient medium. So this is how you have to write, children, with practical number 6. That will be the head. No, don't write any number, just write in a pencil as number 6, because obviously, children, the numbering towards the end that we'll be doing while making the index, we will number them from the beginning in the same serial order for the council to assess our practicals and evaluate easily, right? So till here was 1, 2 and 3, but here please don't write the numbers only. If you want, you can write it for the uniformity in pencil. So this was 4, 5 and 6. Please write the numbers in pencil, because this will change while making the index. Your writing part should be complete, and that's more important. So